Vanakkam. Welcome to this course on classics in neuroscience. In this series of videos, we have been looking at functional organization in neurons and functional organization of dendrites. In this video, we will discuss the concept of dendritic dominance and the seminal contributions of Wilfried Rahl, the physicist turned neurophysiologist. So, the first evidence for properties of dendrites came perhaps from the lab of Eccles, from the work of Eccles, Libet and Young. In their work they showed, using intracellular electrodes, there were small spikes in motor neurons that were undergoing chromatolytic degeneration after cutting or transection of their axons. This gave rise to this notion of specific hot spots within the neurons that were more likely to generate action potentials or spikes or activity. Later, in work led by Eric Kandel, Eldon Spencer and Eric Kandel, they showed that there were small pre-potentials in apical dendrites of hippocampal pyramidal neurons. Yes. These studies, as with the other studies, were conducted in anesthetized animals, anesthetized preparation. But somehow later it was believed for quite some time that dendrites were rather passive membranes, unlike axons, that can conduct and produce action potentials. Only the advent of patch electrode and technology, it came to be shown that there were voltage dependent ion channels in dendrites just like in axons. But the production of action potential, the generation of action potential dependent on multiple things like dendritic geometry, channel density, neighboring channels. So, there is a whole bunch of factors that play, that decide the nature of whether an action potential will be generated or not. Also, in the 1950s, there was the availability of the HH model of conduction of action potential in the axon. Can we come up with a similar model for conduction in a dendrite? That is the question. So, what is shown here is an equivalent model. This of course is an analytical model. Is there a similar quantitative approach for dendrites? The answer is no. So, what was done by Hodgkin and Huxley is a quantitative model or analytical model of action potential in the squid giant axon. And there were challenges in applying this model to dendritic trees. Why? Because there were just so many branches, there were many branches, the number of such circuits will become huge. So, there was a huge complexity due to the branching patterns. Remember about the Purkinje cell neurons, I would have mentioned in the previous set of videos and would have given reference to my previous classes. The Purkinje neurons contain sometimes as high as 500,000 dendrites, very complicated geometry, very complicated number of or very large number of dendrites, and making a model, such an analytical model for each of the dendrite would lead to huge complexity and processing time. It would not be a trivial problem. This is where Wilfried Rahl came into the picture. Wilfried Rahl was born in 1922 and studied physics, and he was part of the Manhattan Project, where he worked in Chicago, and he was introduced to biophysics by Kenneth Cole's program at the University of Chicago. Remember Kenneth Cole from our previous discussions. He was very keen on understanding how the dendrites worked and the challenges that the dendrites provided to understanding how they conducted electricity. So, he was very inspired by the work of Jack Eccles, and so moved to New Zealand to pursue that line of work, and later went on to pursue his own PhD thesis research with Archibald McIntyre in the same university, University of Otago in Dunedin. During his experiments, Wilfrid Rall observed that the potential would spread almost instantaneously or very fast in the cell body and in the axon, but spread rather slowly and in a decaying manner in the dendritic tree. This got him thinking about how or why would this happen. In the 1950s, when Eccles and his group published data that suggested that when there was injection of electric current into the cell body, there was decaying of the response that can be interpreted to be a single exponential, as we would expect from a resistor capacitor or RC circuit with some time constant. Immediately, Wilfrid Rall realized that this could be due to the flow of current in the dendrites. He published this idea as a brief note or a brief communication in the journal Science in 1957, and he was inspired to pursue this line of work and test this idea or test this hypothesis rigorously. So, he performed a whole bunch of analysis, and he was interested in reducing the number of dendritic branches into a single cylinder to analyze. However, all these papers were rejected due to various reasons. This time or this set of experiments and analysis began what came to be called as the dendritic dominance in integrative activity of neurons. This is the notion of dendritic dominance. Dendritic function was modeled by Wilfrid Rall using the input output relations that he suggested. So, he created these kind of models, and he created the idea of equivalent cylinder concept, where he reduced multiple branches into a single cylinder. But like I said, that he faced several challenges in publishing these groundbreaking studies. He was rejected, his papers were rejected multiple times. Finally, he published some of these results in 1959 and 1960. Then, instead of using analytical methods like the ones that HH, that is Hodgkin and Huxley, used, he thought it might be better to use numerical approaches. So, it is important to realize that you need to choose the right method for the system that you are working with. In the squid giant axon, the analytical approach and the model that was developed by Hodgkin and Huxley was appropriate. But in the dendrite case, if you use that, solving of those analytical equations is going to take a really long time. Instead, it would make sense to use numerical methods, that is measurement from multiple places from experimental data, and using numerical methods to solve those. He essentially used a program that was made by Mons Berman, where that was made for a different purpose, for studying radioactive decay. He used a similar model for analyzing spread of activity within a dendrite. He presented his findings in the Ojai conference in California, and finally published this in 1964 and 1967. This was perhaps among the first rigorous presentation of electrical current in dendritic trees. So, there was this non-linear interaction of conductances that underlied synaptic potentials in dendrites. Also demonstrates the specific challenges in incorporating properties of dendrites in neuroscience. There was considerable reluctance among neurophysiologists and neuroscientists in general, neurophysiologists, neuroanatomists, and computational neuroscientists to use this kind of models. But it would be fair to say that with this work, the birth of computational neuroscience as a small subfield of neuroscience happened with the advent of this work by Wilfrid Rall. With this, we come to the end of this video. In this video, we looked at the idea of dendritic dominance and the seminal crucial contributions of Wilfrid Rahl. Thank you for your attention.